So first, I would like to thank the organizer for allowing us to present this work here. It's been a pleasure to listen to all those different views about pathways. So I will present today a new model that we have been developing in gene ontology to tackle some of the problems that we had about representing biological knowledge. And to start with, I just want to point out that indeed Go was founded 20 years ago, so that's a very old project and with also very old habits. But at the very same, our objective never changed. We still want to be able to annotate a specific gene to a specific function. And it's not always easy to state what we mean by specific. And we wanted that knowledge also to be available for computational search and so on, and of course to be open source, open access, both our data and our code. If I have to simplify a lot what is gene ontology, it's obviously an ontology to describe things in a unified manner, and also a set of annotations that are created by a lot of great people that spend a lot of time trying to consistently annotate what are biological function processes and to map them to those terms so we can share that understanding of function and processes. And also sometimes it can even be a kind of philosophical, like when you have a biological process, where does it start? Where does it end? If I have a molecule that regulates that process, is it part of that biological process, or is it only outside and regulating, having some effect on that processes? And obviously biology evolves, so all of our ontology must evolve too. So the ontology itself, you have a quick representation here with a screenshot for a quick go. And you have in black the is a relationship, and in yellow the regulate relationship. And if I take the example at the bottom, we have a regulation of apoptotic process. And indeed, I can say that it is a regulation of cell death. And if I want to be very general, it's a biological regulation. And at the same time, if I follow the yellow links, indeed, because I'm speaking of a regulation of apoptotic process, I'm regulating a process. In this case, an apoptotic one. And we have also terms create and also relation and cross-reference with other databases. And the thing that was important for us also is really to interconnect this data with all possible databases. So that's why we also started to use all. And for instance, this specific term has a unique URI address at the bottom. So anytime you want to refer to that specific process, you just have to say, this is this address. This is its unique identity. And right now, to date, we have 45,000 terms, around 130,000 relations between all those terms, like you can see on the left, and we are linked to 14 ontologies. And I want to stress out that whenever you take the chance, the leap of faith to link your data to an ontology, it's actually very important. Because whenever you do that, not only will you make the world a less chaotic place, but also we are beginning to share and interconnect our data. And by doing so, we increase our understanding of biology. We extend both our understanding and view of biology. So that's very important for us. In terms of annotation, I have an example from a curator that wanted to annotate a human MSH2 gene as having a DNA mismatch repair, and he had some experimental evidence about that. So right now, the way that Go annotation were stored, we have the Go term in green, in blue, sorry. We have the gene with its HGNC identifier in green, sorry. Go term in blue. And we have the evidence with an echo code and a PMID. And right now, all of those, because we're using OL and LDF, are encoded with URI. So whenever you want to refer to the gene, whenever you want to refer to the function, or with the evidence or the article, we already have that. And the very nice thing about that model is like, if I want to have all the genes involved in DNA mismatch repair, that's straightforward. If I want to have all the function of the genes, that's straightforward. And if I want to also say, because Go is agnostic, we are not human specific, we can also find if that function is conserved across species. And just to give you a number, we have today 750,000 annotations curated by our people on over 140,000 articles, which roughly gives 7 million annotations across 3,200 species, so that's a lot. And the important thing, once again, here is we share a unified vocabulary. So whenever you want to compare a function across species, you can. Now the challenges. One of the challenges is, for instance, this Go term, negative regulation of transcription from RNA polymerase II, promoter, blah, 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 involved. You can see that's a very long term. And that was one of the problems of the Go. We have 45,000 terms because we wanted to enable our curators to be as specific as they could when trying to say something. But in reality, in this piece of information, I have three things. I have the fact that I'm talking about a transcriptome for RNA polymerase II promoter that is involved in some processes, the cellular response to UV, and I'm not speaking of the process, but actually of a regulation of that process. So we combine all of that into a Go term, hence a lot of complexity in Go. And also, whenever we have this simple relation between a gene and a Go term, what actually does mean this relationship? Is that involving? That's very general. What does it say? Also, we wanted to be able to annotate not only papers, but biology. And sometimes a curator, because they want to be very specific, very honest, they won't say something because they were only able to annotate a paper. But what we want at the end is really to annotate biology. So we enable also this fact to include as many evidence as required. And the last challenge is like, we really wanted, and this is a very important point for us, to be activity-centric and not gene-centric. Because a gene alone is nothing. A gene, to have a function, needs to have interactions. It needs to be in a context. So we wanted to be more specific about our annotation, and we are taking this paradigm shift in a way to annotate not genes, but activities. So now about the representation itself. So this is basic representation of the gene ontology causal activity models, GOCAMs, shorter. You have the activity at the center in red, and what we are saying is like this activity is enabled by some active entity. Those active entities can be a single gene product, it can be a complex, for instance, and also maybe it has some input, some other participants that are not directly involved in the activity, but are still useful. And maybe it occurs in some location, in the nucleus or transmembrane of whatever, during certain phase of biology, and is already part of some biological process. And furthermore, in addition to that model, we wanted to be able to link activities together, to link biological process together. So that's why we also have those causally upstream of with other activity, with other processes. And once again, we're using OWL, we're using RDF, and our goal was to be able to interconnect with as many databases as possible. So anytime that I'm expressing something, it's actually what we call a triple. We have a subject, a relation, linking to another object. So my subject here could be, for instance, the activity. My relation could be enabled by, and an object could be, I don't know, CDK2, P43, or whatever. And all those relationships are already existing in ontologies, and we're using them. So part of is this long URI enabled by this other URI, and so on. But the beautiful thing about that is not only are we giving unique identifiers to entities, but also to relationship. So it really enables us to share a common vocabulary and way and grammar to say things about biology. So if I take back my example, about which I will not read again, about the negative regulation, on the left, we had the former Go annotation to try to describe that. And on the right, that's the GOCAM models that we have today. We have the net4 that enables some kind of ubiquitin protein ligase activity that negatively regulates RNA polymerase activity. And then we have all those other details to further annotate this function in the scope of its processes. And in addition to that, the fact to be able to use OWL also allows us to use reasoners. And that's a beautiful thing, because anytime you are annotating something, you might do errors. We are not God. We do errors from time to time. Hopefully not too much. But if I create, for instance, this cell proliferation process, I can say that he has a participant, like CDK2 is a gene, and that's fine. But on the right side, I'm actually saying that the cell proliferation process has a participant, which is an activity, and that's not fine. So that was a mistake by the curator, because as participants, the way we defined this grammar was only I have a processes. Well, it can have participant, but the participant mostly can only be gene product or complexes. And this is an activity, so that's not right. And all those checks can be done automatically to help also the curators to try and understand where there might be problems. And also it has been greatly useful for us for doing QA/QC on all of our annotation. And one of the goals of Go is really to transit from the Go annotation to Go CAMs. So we will have hopefully all our Go annotation into the Go CAM format and able to do all of that. So right now we have a website, geneontology.org/CAM. This is how it looks. Basically, we're giving access to the content in terms of biological process, molecular function, and cellular components for each model. So you will have an idea of what contains a GoCam model. And on the right, you will see that you have also access to Sparkle endpoint and REST API. So that's the thing, one again, we wanted this data to be interconnected with other Sparkle endpoint and with the semantic web at large. So this is a simple query, for instance, to retrieve the models and the curators. You can see that we also use your archive, good idea. And yeah, so one of the goals to be here today is also to be able to interconnect our data with everything that has been said, and I think is beautiful. And we had also the presentation of Jalal about the overlap, the Venn diagram between the different pathways. We don't have the same data as you. It's not a competition. But if we can merge all of that knowledge, it's just beautiful. And so my takeaway here is really we wanted to introduce this paradigm shift. We are not annotating genes anymore. We are annotating those activities because we think they are more specific than just saying a gene. A gene in different contexts has not the same function. And in terms of expressivity, the fact to enable those relationships allows us, for instance, to detail the participants, not the ones that are active, but that still require architecturally, for instance, to create a molecular machine, and also to detail what are the units that are actually active, and also to be able to say when and where this activity occurs, and also to link all of those biological functions together to try to make sense a bit more, if we can, about biology. And really, one of the things, the real things that we wanted about that is to be able to ask complex questions from our database, but from all Sparkle endpoints too. So one of the things that I would hope also here today and in the future is that we discuss about how we can improve our interconnection between our databases so we really extend our open access and open source data and strategy. So very last thing, just to give you an idea of how a curator will do it, we have a very simple view, which is called Nutrigraph, where you have the gene products that we spoke about, and then you have the function, the activity. And for the activity, you can see that we have the as inputs, those are the participants. You have the happens during, that's a phase in which the activity occurs. And you can say status also if the activity is part of some process and so on. So we try to do a very, very simple thing for curators because the poor guys are already doing a very complex job. So anytime we can simplify a user interface, that's great for them. And we also have the Nutrigraph interface here, where we can retrieve these examples that I gave earlier about the very, very long Go term. And you can see one activity enabled by a gene with some input, so some other participant occurring in some places, being part of a process, and so on and so on. So that's how we are moving on gene ontology to describe our biological processes and to give more information. And all of those information also have evidence so that we can trace back how and why this model was done. So at the very end, what we now have a DOI version of all of our data that you can access. We have the Sparkle endpoint, the API endpoint. We are open source, so all of our code is on GitHub. And with that, I would like to thank you all, to thank also our PI, Paul Thomas at USC, and Chris Mangan and Susanna Lewis in Berkeley Lab. Seth Carbon, an amazing programmer that might be shy after I say that, in the center of the room, I see you. And Ben Good that will give a talk tomorrow on Biopac's pathways. So thank you all it's a pleasure to be here.
Maybe one question?
I'm not planted. Could you talk a little bit about how you're transforming the hundreds of thousands of annotations that were made in a gene product-centric approach into the new activity-centered?
That's a very good and very complicated question. Thank you. So the thing is, every group that has created this annotation, we spoke about the fact that we don't really know what was intended into that relationship here between the Go term and the gene. So one of the things that we are doing is actively speaking with the different groups to try and see what they had in mind when doing this annotation. For some, it's easier. Like with POM-base, we do have some pretty good idea of what they meant because they use it consistently. For other groups, that's really very difficult, and we don't have straightforward solution except that using a pretty generic relationship, which we might do in the first place. What we are hoping for is, like with the wealth of all the Go annotations that we have, also the ontologies that describe many, many things. Perhaps with Reasoner, we can try to guess what was intended into that relationship. But that's still an open discussion.